Hey guys, welcome to Alevel Biology Demystified, and this time we will be doing the third video on gene technology, and the first topic is PCR, that is the polymerase chain reaction by which we can amplify a small amount of DNA theoretically to infinity, as an infinite amount of DNA can be produced from a small amount, even one molecule. So there are three steps. The first is denaturation, the second is annealing, and the third is complementary strand formation. And this process continues on and on and on and on repeat till the needed amount of DNA molecules are produced from the original amount. So in the first step, we have somehow extracted the DNA we want to amplify. For example, some hair was found on the scene of a crime, and the DNA from that hair wasn't sufficient to carry out the investigations needed. So firstly, the DNA was extracted from the hair and then it was amplified, so a lot of investigations could be carried out on it. Number one, the DNA that is extracted is heated briefly to denature the DNA, that is to break apart the double helix. This is the first step. And the second step is when something called a primer is added to both of the separated strands. And what is a primer? A primer is basically a short length of DNA, often about 20 base pair long, and it has a base sequence that is complementary to the start of the part of the DNA that we want copied. And why do we need to add these primers? It's because the DNA replication process by DNA polymerase cannot begin without any sort of guidance. So the primer is basically telling the DNA polymerase to come and do its job right over here. And this is the annealing process. So after the primer attaches to the start of the DNA over here and over here, because replication can only go in the 3' to 5' direction, so the start of this DNA molecule will be over here and the start of this DNA molecule will be over here, because we can only replicate from 3' to 5' direction. So once the DNA has been copied, right, the mixture is heated again, and which once more separates the two strands in each DNA molecule, leaving them available for copying again. And so this process continues on and on and on. And once more the primers will fix themselves to the start of each strand of unpaired nucleotides, and the DNA polymerase will come up and make complementary copies. So the three stages operate at different temperatures. So the first part right over here, in which the DNA molecule is denatured, takes place at a really high temperature of 95 degrees Celsius. And then comes the second step, in which primers, that is short lengths of DNA, often 20 base pair long, that have base sequences complementary to the start of the DNA that we want copied, is introduced. Actually, it was introduced in the beginning, but now that the base pairs are exposed, it is capable of binding. And the annealing process basically takes place at a temperature of 65 degrees Celsius, so the temperature has to be cooled down somewhat for annealing to take place. And then for the DNA polymerase to come up and use free nucleotides to synthesize a complementary strand, it needs a temperature of about 72, which is the optimal temperature of a particular version of DNA polymerase called Taq polymerase. And this process is actually called elongation. So we have denaturation, annealing, followed by elongation. And the temperatures are 95 to 65 to 72. And Taq polymerase, which is used for this process, actually comes from microorganisms that have evolved to live in extremely hot environments like heated vents. And most laboratories that work with DNA have a machine that automatically changes the temperature of the mixture. The DNA sample is placed into a tube together. So in the beginning, all of the stuff that is needed is put in. That is the primer, the free nucleotides, the buffer solution, and DNA polymerase. So something that was not mentioned before is that a buffer solution is also needed. And the PCR machine is switched on and left to work. And the tubes are very small and have very thin walls. So that when the temperature in the machine changes, the temperature inside the tube changes very quickly because the tubes don't have a lot of capacity, and in addition the walls are very thin. So heating denatures the DNA, which starts a new cycle of copying following steps 1 to 4, and repeating the cycle 10 to 12 times copies the gene many times because each time more copies are made, and that is why we call this a chain reaction. What is the difference between a primer and a probe? They're both short sequences of DNA. However, whereas a probe is used for identification and separation purposes, a primer is used to initiate the production of a complementary strand for a DNA template to which the primer is attached. So they are basically the same, but they have vastly different functions. But fundamentally, they are the same thing. The function they serve is different because they are used in different circumstances under different situations. So, from this, there are some things you need to remember. That replication and complementary strand synthesis both only take place in the 3' to 5' direction, and that BCR has 3 main steps: denaturation, annealing, and elongation, and each of them take place at different temperatures at 95 degrees Celsius, 65 degrees Celsius, and 72 degrees Celsius. And that all of the stuff that is needed is added at the beginning to a small tube which has a very low capacity of about 0.05 centimeter cube, and things that are added are basically something called a primer, free nucleotides, buffer solution, Taq polymerase, and the sample of DNA which we want amplified, and this is put into a machine that automatically changes temperature for each stage. And a primer is basically a guidance, and it binds to the start of the DNA molecule which we want a complementary strand for, and that is it. tag polymerase will not have to be added again before the elongation process, because the one that worked in the previous elongation process and the one before that was still in the solution and ready to do its thing. So it is not destroyed by the denaturation step, so it does not have to be replaced during each cycle. This is the first reason, and why I'm telling you this, because I've actually written tag polymerase in the syllabus, so I was thinking they may give this as a question. They were saying you have to really emphasize the role of tag polymerase. And number 2, because it operates at a high temperature, meaning it is a high optimal temperature, means that the temperature for the elongation step does not have to be dropped below that of the annealing process. So efficiency is maximized, otherwise we would have to cool it down and provide heat again, and that would have required a lot of power, but we don't have to drop it below the annealing process. We can actually maximize, we don't have to cool it down and then provide heat again, which would have required a lot of power and would have taken some time for the right amount of temperature to be achieved after the whole solution was cooled down for the elongation process. So has a high optimum temperature, which means that the temperature for the elongation step does not have to be dropped below that of the annealing process. So efficiency is maximized. And what do we use PCR for? To amplify DNA from the smallest tissue sample left at the scene of a crime, and this has helped solve many different cases. How many molecules of DNA are produced from one double-stranded starting molecule after 8 cycles of PCR? So 2 raised to power 8. You will get approximately 256 double-stranded molecules from 1. So the formula for this is 2 raised to power n. Really interesting if you ask me. Explain why it is not possible to use PCR to increase the number of RNA molecules in the same way, because RNA is single-stranded. So this just doesn't work out. You have one single-stranded molecule, this is RNA, and you cannot make a complementary molecule for it. RNA basically exists as a single-stranded molecule, and you cannot make a complementary strand, and because you cannot make the complementary strand, you cannot use the complementary strand to form another complementary strand following denaturation. It's really simple, just a bit difficult to verbalize, so to speak. So they ask you for 2 marks because RNA exists as a single-stranded molecule and it does not have a complementary strand and cannot form complementary strands, and that's literally the basis of PCR. And now we actually come on to something called microarrays, and they are really important for 2 reasons, and they are very important reasons, and they're actually mentioned in the syllabus. The use of microarrays in genomic analysis and in studies of gene expression, because when you use them you can figure out which particular genes are present in the genome, and also to show the level of gene expression of a particular gene in a particular cell. So what is a Gaussian microarray? Well, they have helped us to identify the genes present in an organism's genome and to find out which genes are expressed within cells and how much expression is there. And they've also allowed us to study a very large number of genes in a short period of time, and so the information available has been really increased. But what is a microarray is the question of the hour. It must be something really cool and really complicated and really badass for us to have gotten so much from it. Well, yeah, it is badass, but it isn't complicated. Not at all. So, a microarray is a small piece of glass or plastic, usually about 2 centimeters squared in area. So you can measure how much 2 centimeter cubed is with a simple ruler. And short lengths of single-stranded DNA are attached to the support in a regular 2-dimensional pattern with 10,000 or more different positions per centimeter cubed. So when they ask you what on earth is a microarray, we say that it is a small piece of plastic or glass, usually 2 centimeters squared, to which short lengths, and short is important here, to which short, and you need to underline this so you don't remember, so don't forget, to which short lengths of single-stranded, again important, obvious but important, single-stranded DNA is attached, right? And how is it attached? In a regular 2D arrangement. And you can also add this in the definition that there are at least 10,000 or more different positions per centimeter cubed, per centimeter squared of the microarray to bring into notice just how many probes are there in one very measly, one very small microarray. And each individual position has multiple copies of the same DNA probe. This is also important. So, 10,000 or more different positions per centimeter squared, and you need to remember positions here. This is actually a key term here because position means a position has many different identical probes, another position will have many different from the previous one anyways, many different but the same to each other probes, and each position has multiple copies of the same DNA probe. Alright, hopefully you're getting a hang of what all the microarrays are all about. So if you want DNA probes, you can search databases to find DNA probes for a huge range of genes. So having selected the gene probes required, you want this and you want that, an automated process applies these probes to the positions on the microarray. So you can basically customize microarrays for your use. So you can write, it is possible to customize microarrays for whatever use you want. When microarrays are used to analyze genomic DNA, which is the first part of this topic in the syllabus, The probes are from known locations across the chromosomes of the organism involved and are 500 or more base pairs in length. And a single microarray can even hold the probes from the entire human genome. And remember, while primers were about 20 base pair longs, and probes that were used in gel electrophoresis were also 20 base pairs long, the probes that are used here are 500 or more base pairs in length, because you want to be able to differentiate alleles from each other. Even though they are mostly the same, they do have differences. So this might be the reason why we want bigger probes. So microarrays can be used to compare the genes present in two different species. Alright, we have two different species. We have a horse and we have a donkey, and we want to be able to compare and contrast the genes that are present, the genes that both of them have, the gene that one of them has but the other does not. So DNA is collected from each species. From all of the DNA, perhaps from each species, is collected and is cut up into fragments and denatured to give lengths of single-stranded DNA. And why is it denatured? Well, obviously so that it can form complementary bonds with the probes on the microarrays, otherwise it won't be able to do so. And it is also labeled with fluorescent tags so that we know if it is binded or not. For example, the DNA from one species may be labeled with green tags, and the DNA from the other species could be labeled with red tags. And the labeled DNA samples, they are mixed together both from both the species, and then they're allowed to hybridize, and that is a key word, hybridize with the probes on the microarray. And any DNA that does not bind to the probes is washed off, right? And the microarray is then inspected using UV light, which causes the tags to fluoresce. Where this happens, we know that hybridization has taken place because the DNA fragments are complementary to the probes. Green and red fluorescent spots indicate where DNA from one species only has hybridized. Where DNA from both species hybridizes, a yellowish color is seen, which is the result of both the red and yellow being really close to each other, and so our eyes see them as being yellow. So yellow spots indicate that the two species have DNA with exactly the same base sequence. This suggests they have the same genes. If there is no color for a particular position on the microarray, it means that no DNA has hybridized with the probe and that a particular gene is not present in either species. So let's go over this again. The use of microarrays in genomic analysis. So when we're doing this, when we're doing genomic analysis, what happens? That probes are from known locations, known as in you know where you're taking the probes, are from known locations across the chromosomes of the organisms involved. For example, if you are comparing the DNA of two humans, so insulin gene might be taken, the gene for the CFTR protein might be taken, or the probe for the CFTR gene might be taken, and the probe for the insulin gene, and which allele you take the probe for is on you, depends on what you're checking. Probes are from known locations across the chromosomes of the organisms involved. And another important thing is 500 or more base pairs in length, and we're talking about the probes here. And the reason I'm repeating all of this again is because this is a bit difficult to remember. So repetition does really well in cementing ideas into your head. And what can microarrays be used for? They can be used to compare the genes present in different species. So genomic analysis includes comparison of genes found in two different species. So what are the steps? Step number 1, DNA is collected from each of the two species. The DNA is then fragmented and the DNA is then denatured to give lengths of single-stranded DNA. And lastly, the DNA is then labeled with fluorescent tags, that is labeling with fluorescent tags. And each DNA is labeled with a different tag. The DNA from one species might be labeled with a red tag, and the DNA from the other might be labeled with a green tag. And in step E, the DNA samples are mixed together, that is from both the species are mixed together. And in step F, they are allowed to hybridize with the probes. Hybridization with probes is technically the last step. And now comes step G, which isn't really part of the process, is that unbinded DNA is washed off, and then comes H, which is actually the inspection part, is that the microarray is inspected with UV light. So H is inspection with UV, HI. Right, when this happens, we know hybridization has taken place because DNA fragments are complementary to probes. Green or red indicate that DNA from one species has only hybridized. Where both species DNA is hybridized, you would see a yellow color, which is a hybrid of red and green. And where there are no fluorescence from a position, it means that neither species contain this particular gene, and that a particular gene is not present in either species. And then comes the use of bioinformatics in microarrays, and we'll actually have a separate topic for bioinformatics later. The microarray is then scanned so that data can be read by a computer. Data stored by the computer indicate which genes are present in both species, which genes are present in one species alone, and which are present in neither of the species. So in step number I, microarray is scanned so that data can be read and also stored by a computer. Can be read, and in brackets you can write processed plus stored, and that is basically bioinformatics. And it looks really pretty, doesn't it? The microarray, which is fluorescing. So this position is green, meaning both the species contains it. This is red, meaning one species contains it. This is yellow, which means that maybe all of them contain it. and the colors are analyzed to show which genes or alleles are present. Right, and the second thing we can use microarrays for is to check which genes are being expressed at any specific time in any cell in the body. For example, the genes that would be expressed by a cancer cell would be really different from those in an active but non-cancerous cell. And how can we use a microarrays to tell which genes are active and which are not? We can check for the gene products, that is, for the mRNA. So the mRNA from the two types of cell is collected, right? So if we want to see if gene expression is happening or not, and because mRNA is itself incapable of the whole binding process, we'll have to collect the mRNA from the two types of cells, and we will have to convert mRNA into cDNA, again using reverse transcriptase. So these enzymes are really handy, they come in really handy. And as the quantity of mRNA in a cell at any one time is small, we may increase the quantity of cDNA using PCR, and now PCR comes into play again. Now please, I'm hoping you really get the importance of all of these processes and all of these enzymes, because they're used again and again. So PCR might come into play, and the cDNA, like before, is labeled with fluorescent tags. It is then denatured to give single-stranded DNA and then allowed to hybridize with probes on the microarray. And the spots on the microarray that fluoresce indicate genes which were being transcribed in the cell. And the intensity of the light emitted is an indication of the level of activity of each gene, because if there was greater activity, there would be more mRNA produced. And because PCR amplifies all of them by the same amount, the ratios remain the same.